Now we're going to have a virtual demo to learn and explore GlyGen.org, a data integration and dissemination resource for glycoscience funded by NIH Glycoscience Common Fund. The demo will showcase how to use GlyGen in your research and to find answers to complex questions like what kind of glycans have been reported to be linked to specific glycosylation sites on my glycoprotein of interest in a single resource, GlyGen.
Hello, everybody. Thank you for joining for that live demonstration. My name is René Ranzinger from the Complex Carbohydrate Research Center, and I will make this demo together with Nathan Edwards from the Georgetown University. We are going to show you GlyGen, a web portal which integrates glycomics and proteomics data. This resource is funded by the NIH Common Funds program, and the aim of that program was to integrate glycomics with other fields of research, for example, proteomics. As you can imagine, that's a really big task to connect glycomics databases with other databases in proteomics, genomics, and so on. There's a really big team working on that, in addition to the people at the Georgetown University, George Washington University, and the University of Georgia. We have a lot of international collaboration partners from the glycomics databases, for example, Kyoko Aoki Kinoshita, Nikki Paco, Matthew Campbell, but also from the proteomics side. So we are in contact with Amble EBI and Uniprot, and we are also working closely with the NIH and CBI folks. So how does this work? Before the grant even started, we made meetings and asked researchers in the glycomics field and also beyond in the proteomics field, what are the kind of questions you would like to answer with using databases that you cannot answer right now because these databases are not connected. And they provided us with a very long list of these questions. And based on these questions, we selected many resources, glycomics, proteomics, genomics, and so on, that we needed data from to be able to answer these questions. And in addition to these resources, we also have data from people that generate data and are willing to share this with us. We take all this data, we download this information from these resources, and then we standardize the data, we deal with the different data formats, we do some quality checks on this data, and then we integrate that into one consistent data collection. Of course, that data collection is only useful if people can actually access that. That's why we provide a lot of different interfaces where people can get access to that data. Some of these interfaces, like our Sparkle endpoint or the web service API, are meant for computers, bioinformaticians to, download a mass of data. But we also have interfaces that normal users can access using any kind of web browser, the data portal and also our web portal. And in this demo that we are going to do now, we are concentrating on these resources that everybody can access. The data is freely available. Anybody can go, just type this URL in your browser and you have access to the data. It's freely available. Download it, do with it what you want. The only restrictions on the license is that if you use that data, you have to attribute where you got that information from. But besides that, you can do anything you want with it. Let's start with the demo. So this is the web browser. If you type this URL in your browser, that's the web page you will be starting with. It shows right here the main information that we deal with where are the glycans, the proteins, and of course the glycoproteins, which connects the glycan and the proteins, or it's one way to connect the glycan and the proteins. You have also a bunch of other information on the front page, statistics that shows you how many glycans and proteins we have. As you can see, we are concentrating mainly on human, mouse, and rat right now, but we are expanding the scope. Due to current events, we started integrating information about the coronavirus and also hepatitis C. What we also have on the front page is our Twitter feed. So if you would like to get updates, we are mainly using the Twitter feed, just subscribe to it. Okay, let's get started with the glycans because you mainly care about the data. So the easiest way to start with glycans is just click on that box and it will bring you to the glycan search page. On pretty much every page where we have forms, we always provide little examples that you can easily just click on and get started, just in case you don't know how to use that system in the beginning. This box essentially allows you to drop any kind of information that might be relevant to glycan and just search for it. For example, this, what we are seeing right now, is a glytucan ID. It would find me that corresponding glycan, but I can also look for an enzyme, for example, ST6-GAL1, which adds sialic acid to N-glycan structures and just see what the system finds with it. So I'm clicking on search and it presents me with a list of glycans that are annotated with that enzymes, where that enzyme may have been involved in the synthesis of that glycan. You see the glytucan accession number, which is a unique accession number for every type of glycan. A graphical representation, the mass, and some other information. On pretty much every data page, you also have a download link, which allows you to download the information that you see and do some post-processing on your local computer. For example, this table you can download into an Excel spreadsheet and then do something on your computer. Another thing I would like to point out, here on the right side, you see that little widget feedback. On pretty much every page, you can provide us with feedback. So if you see something on a web page that doesn't make sense, or you think there's a mistake, or if you have any kind of other information you want to share, just click on that. Feel free to type a message, leave your email, and we will get back in contact with you. Okay, coming back to our glycans. So there's a bunch of glycans that may have been made by this enzyme. By clicking on the glytucan accession, we can actually open the web page, which shows us detailed information about that glycan. So it's divided into these little boxes. In the first box, the general box, you see the image again. We have the glytucan accession, the mass, monoisotopic and permethylated, the composition, different types, and some other accessions. In the next box, we see the species annotation. So this glycan has only been annotated with the human species. It might be present in other species as well, but the only evidence we could find in the databases that we integrate is human in that case. And you can also see which databases have provided that annotation. So in this case, Glytucan and also Unicarb have provided that annotation. This is always indicated by these badges. Now, by clicking on that badges, it will expand a list of accessions that have provided that annotation. And if you click on these accessions, it will open a new browser tab for you and bring you to the web page of that glycan in this database. And here you can find additional information. So when I said in the beginning, we download that information, we don't really download all the information from all these databases. We only download a core information that we consider important and integrate them. But using these links, you're actually able to go back to these resources and get additional information. So GlyGen is really like a hub where you find your glycan or your protein, and then you have a lot of links out to these other resources which have additional information. Okay, so this glycan has been found in human. The next thing is a list of common motifs that are found on that glycan, a list of glycoproteins that have been reported to carry that glycan. I will come back to that in a second. Then we have a list of enzymes that may have been involved in synthesizing that glycan, potentially in the different species. So there are some mouse enzymes, and there will be also some human enzymes later on. We have digital sequences. We try to support as many as possible because you might be using them in other software tools or other databases as an input. And we have the cross-references to other databases where this glycan has been found, several CardBank references, CABI, GlyConnect, glycosciences.de, CAG, PubChem, Unicarb, and so on. And all of these databases might provide additional information, and you can get to them right from this page here. And then in the end, we have a bunch of papers where this glycan has been reported. Let's go back to the glycoproteins, because that is the main aim of that resource. Connect the glycans with the proteins and make it easy to, browse that information. Here we can see that there are a bunch of proteins where this glycan has been reported. We have the UniProt accession number and the position where that glycan has been reported. And again, the databases, or in this case for PubMed, the papers that provided that annotation. Now, by clicking on the UniProt accession number, I'm going to the protein page of that protein in GlyGen. The protein page is pretty much has the same structure than the glycan page. We have that general box where we have some general information like the gene name of that protein, the chromosomal location if it's known, the UniProt accession number, the UniProt ID, some common name for that protein, the mass, and the RefSeq annotations. Then when we scroll down, we can see, okay, that's a human protein. Again, it was coming from UniProt, that information. Then we have some function information, also from UniProt and from RefSeq. The Go annotations are for that protein, the glycosylation table. Here again, we can see what is the glycan that has been reported. Some of them are actually compositions, some of them are real structures. We can see the amino acid, the position where it's glycosylated, and the glycan accession. And if I would click on that, I will go back to the glycan page for that particular glycan. And again, also the evidence where we got that information from, or which database we got that information from. We also have a bunch of sites where no glycan is reported. For example, we have predicted sites, or we have just reported sites where people have written papers that on this particular asparagine there is a glycan because we saw the scar in the MS, but we don't have any idea what type of glycan was on there. Okay. So we have the sequence information. I will skip over that for the moment. We have information, pathway information, in which pathways that protein may have been involved. We have a list of isoforms for that proteins. We also have a list of homologs in mouse and rat for that protein. Then we have disease annotations where people have reported that that protein was involved or changed in some kind of disease. We have a mutation, common mutations that have been reported, and then also in which type of disease that mutation has been reported. We have expression information for the different tissues, if a protein was expressed in this tissue or not. We also have expression information in different diseases, if a protein was over or underexpressed in that disease. Then the cross-references to other databases for that protein. So you can see a lot of common databases. So there's Reactome, the CAG pathway database, a bunch of PDB entries. The UniProt link was right on top of the page, and there are tons of other databases that we link to, which again may have additional information that might be useful for you just to learn more about that protein. And then in the end, we have the list of publications that talk about that protein. And on all pages, again, we have that download option where you can download that information in a data file and then use it on your local computer. Let's go back to the search interface. As I showed, you can start them on the front page by clicking on one of these cards, or you can use the menu and then go, for example, back to the glycan search. In addition to the simple glycan search that we have seen before, which just have a text field where you can drop any kind of information, we also have an advanced search, which allows you to be more specific. For example, you can just provide a glycan ID, for example, like toucan, or you can provide a mass. For example, if you are doing mass spec and you just want to know which glycans have a mass between 2000 and 2500 Daltons. You can also check for the permethylated version of that mass. You can also look for certain glycan types, N-glycans, or glycans that have certain motifs, for example, that are complex N-glycans. And you can also look for glycans just by the specific enzyme, which is essentially what I did in the beginning. You can also make a combined search. So you can say, I want to see all glycans which are between 2500 and 3000 Daltons that are N-glycans and have a certain motif. If I click on that search, I will find that subset which fulfills all of these properties. Another way of searching for glycans is by using the composition search. Here you can specify a bunch of monosaccharides that your glycan needs to have, and it will find all glycans in our database which have that composition. If you need help, you can always contact us, but we also have that tutorial page where you find a bunch of slides and videos that help you working with that, how to use that search page. A similar page we have for the proteins, where again it starts with a simple search where you can just drop anything you like, or you can go to an advanced search where you can provide specific information and make your search more fine-grained to find easier what you are looking for. And here also you have a tutorial. Another way how you can use that system is by using a quick search. A quick search is essentially a complex query that is already pre-prepared for you. It only asks you for one piece of information and then answers this query. For example, which proteins are shown to bear the glycan and on which side is the glycan attached. So if you're just dropping a glyctoucan ID here for that particular glycan of interest, it now will make a complex search where it looks for the protein and the sites and then compiles you the answer to that question. There are a lot of other queries on that web page here that you can use to hopefully find what you're looking for. In case you don't, because you cannot make the query you want to do, always use the feedback, contact us, and we will be happy to help you either, providing you with the answer you're looking for, or by actually making a new quick search that might help you to get the answer you're looking for. So that is a quick overview about the page and what you can do. And now I will hand over to Nathan Edwards, which will show you some more examples how you can use the system and also how you can how other systems link back to GlyGen.
Thank you, Renee. I'm going to focus somewhat more on the interconnectedness of GlyGen with other resources and try to convince you that there is significant utility in especially linking through the proteins to other resources that have been developed over the years. So in particular, I'm going to start by looking for a protein gene name, and I'm going to look for a gene name using the global search function to look for interferon gamma receptor number 1, which is a protein that I have prior knowledge of. When I put that gene name into the search field, I'm doing a global search over all of the GlyGen databases, and I guess I typed it wrong. IFNGR1, interferon gamma receptor 1. Let's try again. Here we go. All right, so now we've done a single search using this gene name across the entirety of the GlyGen database, looking in the glycan section, the protein section, and the glycoprotein section. Since this was a gene name, even though I didn't tell that to GlyGen, I'm going to click on the gene selector here under proteins to look at the three protein entries which match that, that gene name. And I'm going to quickly cut to the chase and select the human version of this protein. As we already saw from Renee, there's a lot of information on this page. I'm going to concentrate on a couple of things. First, I'm going to point out that we have the RefSeq accession here, and we could certainly click on this and go over to the RefSeq entry. In fact, I did that earlier, and if I scroll down, you can look at all of the information in this RefSeq entry. But in particular, notice that we have a link back to GlyGen from the RefSeq page. In general, we're working really hard to integrate information from other resources, but also to provide links, provide information to those other resources through which they can link back to us. I'm going to go down and look at the glycosylation section because I'd like to show you a little bit about what's here in terms of the site information. So one of the most convenient ways of understanding what's happening at a particular glycosylation site is to click on the site number here. Asparagine 79, for example. This takes us to this site view page. If we scroll down, we can see that at Asparagine 79, which is the glycosylation site that has each of these annotated glycans, we can see just the information that's at this site. We can see the information on the sequon, and we can conveniently jump to other sites of the glycan of the protein to see what glycans are present at that site. Nevertheless, I'm going to focus on the asparagine 79 because I want to show you something at the protein level. If I'm still in the sequence view. Okay, if I scroll back down to the glycosylation area, I can then look at the next section, which is the protein sequence. We have a nice graphical view that allows us to see all of the information on the protein sequence and what might be at a particular place. In particular, we can cursor over some of these markers to see what they represent. But in particular, I'd like you to focus on this region over here where it looks like we have a green mutation kind of close to these glycosylation sites corresponding to annotated glycans. So again, I'm going to go back down to the sequence and I'm going to look at the same information in a slightly different way. I'm going to click on the N-link sites marker for the sequence so that we get these little red underlinings under the asparagines. I'm then going to click on the mutation selector, which highlights mutations where they have been annotated on this protein sequence. Notice that we have a annotated mutation right on the serine that's part of the sequon for that asparagine 79. At this point, we can say, I wonder whether or not that mutation is going to disable this N-glycosylation site. We already know that this site has N-glycans attached. Perhaps it's relevant to disease or to mutations. Clicking down here to the mutation section, we can find very quickly that the position 81 with the serine on it has been observed as a mutation in colorectal cancer. And perhaps we might come up with a hypothesis that this is then the glycosylation event or the loss of glycosylation events on that site due to the change in the N-glycosylation sequon is somehow involved with colorectal cancer. I also want to point out that these accessions that we're using for glycans are also observable in PubChem. If you do a search for one of these glytucan accessions, you end up on the search page. You can find it in PubChem. And if you scroll down, you can see that the glycan links are also available for looking at here. All right, I want to do one more quick search. And again, I'm going to use the search bar at the top. I'm going to look for SARS in order to find the entries that we've made recently for the coronavirus proteins. I did a global search for SARS, and it hit in a bunch of places, but I'm going to focus in the most appropriate place, which is glycoproteins and species. There are 4 entries that match the term SARS. I'm going to click on the 4, scroll down, and I'm going to focus on the spike protein. Here's the spike protein. It's given the gene name S, and clicking on it, we get to see all of the information that we have available for this protein. Scrolling down to the glycosylation section, we see the annotated N-glycans on this site. In particular here, this is a very recent paper out of the Crispin lab that has been annotated by Unicarb KB. In particular, this was a mass spec-based experiment. And so the annotations here are glycan compositions. GlyGen is able to represent compositions and structures where they're available, and to make that clear. All right. I'm quickly going to show you a little of the information in the data section. This is information that you can use as an informatician. All of the information that GlyGen consumes to construct the site are listed here. And in particular, I'd like to show you the data that's been acquired as part of the GPT Wiki project that Dr. Goldman talked about earlier today. This is an example of how you might provide your glycopeptide or N-glycan, O-glycan annotations to GlyGen in order to have them integrated with the infrastructure here. It's a very simple format: uniprotic session, glytucanic session, or composition string, position on the protein, etc. GlyGen also provides public APIs. These APIs are the same APIs that the website is built on top of. So if you want to consume this data using these APIs, they are public, and you can use them in the same fashion that we are using them. But you can also design your own queries. We provide a Sparkle endpoint for looking at the information in GlyGen. You can essentially write your own database query against the GlyGen information and pull back whatever version of information you need to get stitched together in order to write your own infrastructure or to consume it in whatever way makes sense to you. Lastly, I'd like to show you that we have a how to cite area of our website. It's very important for this resource to be able to track when people are using it and how it's been useful to the glycoinformatics and glycoproteomics communities. We encourage you to cite the paper and the website if you use it in your work. This how to cite page lists our current primary publication that you can use to cite the publication, and information about how to cite the website itself. All right, I believe I've stayed almost on time. I'm happy to, we're happy to take some questions if we can from those in the chat. Otherwise, please email us from the, you can contact us on the GlyGen site. You can also send email to any of the people involved, including Renee and myself, and we'll be happy to help you with questions.